Welcome to the deep dive. Our listener has sent in a ton of stuff on DNA technology,
Oh,
like
wow.
textbook chapters and research on all the latest techniques.
That's great.
Yeah, we're talking lecture notes and diagrams,
Nice.
even a whole research article. The works. So get ready to decode the secrets of DNA sequencing and genetic engineering.
Right.
And lucky for us, we have our expert here to help break it all down.
Well, think of it this way. It's like we're exploring the instruction manual for life itself.
I love that.
Okay.
Okay, so let's start with the basics.
Okay.
DNA sequencing. I kind of have this picture in my head
Uh-huh.
of scientists hunched over microscopes,
Yeah.
you know,
Yep.
painstakingly deciphering the code letter by letter.
Right.
Is that at all close?
It actually used to be a lot like that.
Really?
Yeah. Early methods like Sanger sequencing,
Oh.
they were revolutionary at the time.
I bet.
But they could only handle a few hundred base pairs at once.
Wow.
Like imagine reading an entire encyclopedia,
Oh, God.
one letter at a time.
That would be
It-
rough.
Tedious.
Yeah. How did we get from that to sequencing entire genomes?
Well, that's where next generation sequencing comes in.
Oh,
It's like going from a magnifying glass to a supercomputer.
huge leap.
Huge. We're now talking about sequencing millions, even billions of DNA fragments all at the same time.
Hold on. How is that even
It-
possible?
Okay, so picture a machine that reads the DNA code,
Uh-huh.
like a ticker tape flashing different colors for each letter.
Oh.
So that's basically sequencing by synthesis.
Oh.
Each flash represents a different nucleotide, like A, T, C, or G being added to the DNA strand.
So it's like a high-speed, multicolor light show,
Exactly.
revealing the secrets of our genes.
Exactly. And by analyzing the patterns of these flashes, scientists can piece together the whole genome sequence.
It's like solving a giant jigsaw puzzle, but with billions of pieces.
Yeah.
That's wild. Okay, so we've got this incredible ability to read the code of life,
Right.
which I guess has naturally opened up possibilities for manipulating it.
Yeah.
And that's where we get genetic engineering. So one key technique is gene cloning,
Yes.
which is basically hitting copy and paste on a specific DNA segment.
It's like making a genetic Xerox machine.
I like that.
So let's say you have a gene that codes for a valuable protein,
Okay.
insulin.
Right?
Scientists can take that gene and insert it into a bacterial cell,
Uh-huh.
essentially hijacking the bacteria to make huge amounts of that protein.
So we're turning bacteria into tiny factories.
Basically.
Pretty clever.
Yeah, but it's not just for insulin production.
Right? Okay.
Gene cloning can be used in agriculture
Oh, yeah.
to modify crops to resist disease or tolerate herbicides.
Makes sense.
Yeah, it's
Yeah.
a powerful tool with so many applications.
Totally. So we can copy genes.
Right.
But then how do we make sure that those copied genes actually do their thing inside a cell?
Well, that's where things get even more interesting.
Right.
Just having the gene isn't enough.
Oh.
It actually has to be expressed,
Okay.
meaning the cell has to read the gene's instructions and make the right protein.
Got it. So I know our source material mentions expression vectors.
Oh, yeah.
What are those?
You can think of them like tiny USB drives
Okay.
carrying genetic information from one bacteria to another.
Interesting.
Yeah, so these are specialized plasmids,
Right.
and they're designed to make sure that a gene is not only copied,
Uh-huh.
but also turned on in the host cell.
So it's like giving the cell the instructions
Yes.
and the raw materials.
Precisely. It's really a fascinating dance
Yeah.
between the copied gene and the machinery inside the cell.
Cool.
And scientists are always trying to optimize this process to
Yeah.
make sure the cell produces as much of the protein as possible.
Makes sense. Well, this is all making sense, but it also brings up another question.
Okay.
How do scientists figure out where and when genes are active?
That's where things get really complex. I
Okay.
bet it's not just about a gene being there, but about how active it is.
Right.
Imagine a city with thousands of lights.
Okay.
Some are bright, some are dim, some are flickering on and off.
I see. So are genes kind of like those lights,
Yeah,
switching on and off depending on what the cell needs?
exactly. And scientists have these amazing techniques
Oh, wow.
to actually visualize this whole genetic light show.
That's so cool.
Like one of these techniques is called in situ hybridization,
Okay.
and it lets us see which genes are active in a specific tissue
Wow.
or even a single cell.
So we can literally see which genes are turned on
You got it.
at a given moment.
That's right.
That's incredible. And then there's another technique called RT-PCR,
Right.
which lets us measure the amount of messenger RNA
Yes.
or mRNA for a specific gene.
Exactly.
And since mRNA carries the genetic blueprint from DNA to the protein-making machinery of the cell,
Yeah.
the amount of mRNA tells us how active that gene is.
You got it.
So it's like we're listening in on the conversation
Yeah.
between DNA and the cell's protein factories.
That's a great way to put it.
Pretty amazing stuff. But what if you want to study gene expression on a larger scale, like looking at thousands of genes at once?
Well, then you're talking about some really high-tech stuff.
Okay.
So imagine a grid with thousands of tiny spots,
Uh-huh.
and each spot represents a specific gene.
Okay.
This is what's called a microarray.
Hmm.
You apply this fluorescently labeled sample of mRNA to the microarray, and the spots that correspond to active genes, they light up.
Oh, wow.
It's like looking at a genetic heat map.
So the brighter the spot?
The more active that gene is in the sample.
Got it. And is that the most advanced way to do it?
Oh, no, we can actually go even further with RNA-seq.
RNA-seq.
Instead of using those pre-selected spots, RNA-seq sequences all the mRNA molecules in a sample.
Oh, wow.
So it gives us a complete picture of gene activity.
So it's like upgrading from a blurry snapshot to a high-def panoramic view.
Exactly.
Very cool. Okay, so we've gone from sequencing single genes to entire genomes.
Right.
From copying genes to understanding how they're expressed.
Yes.
And that brings us to another groundbreaking technology,
Let's hear it.
gene editing.
Gene editing is like having a molecular scalpel.
Oh, wow.
It lets us make very precise changes to DNA sequences.
That's amazing.
And one of the most exciting tools in this field is CRISPR-Cas9.
I've definitely heard of CRISPR.
Yeah.
It's the one that everyone's calling a game changer, right?
Absolutely. Imagine being able to correct a single gene mutation
Right.
that causes a debilitating illness.
Yeah.
That's what CRISPR allows us to do
Wow.
with incredible precision.
And it actually works.
It's like we can rewrite the code of life to erase genetic error.
That's a powerful concept.
It is.
And I can see why it's generated so much excitement.
Yeah.
But I also imagine that there are a lot of concerns about a tool this powerful.
Oh, for sure.
Like, what are some of the ethical concerns that keep you up at night?
One of the biggest ones is unintended consequences.
Okay.
The human genome is incredibly complex,
Right.
and we are still just learning about how genes interact with each other.
Yeah.
So even a small change to a single gene could have ripple effects throughout the genome.
It's like the butterfly effect,
Sure.
where one small change can lead to a ton of unpredictable changes.
Exactly. And then there's also the issue of germline editing,
Okay.
which involves making changes to the DNA in embryos or reproductive cells.
Right.
And those would get passed down to future generations.
Yeah, that brings up a whole other level of ethical dilemmas.
Right.
We're talking about changing the genetic makeup of future humans
Yeah,
who have no say in the matter.
exactly. It's a huge responsibility, and we need to approach it carefully.
For sure.
Where do we draw the line? Who decides what traits are good and which ones are bad?
Right.
These are all questions that society needs to figure out
But-
before we go too far down this road.
Well, this has been an incredible journey so far.
It has.
We've gone from the super slow process of early DNA sequencing
Yeah.
to the mind-blowing possibilities of gene editing.
It's really incredible.
It's clear that DNA technology is revolutionizing science and medicine.
And we're just getting started.
Oh, I like that. Stay tuned, because we're going to explore some real-world applications of all this technology in the next part of our deep dive.
That's right. We'll see how DNA technology is already being used to transform things like medicine forensics, even agriculture.
Can't wait. Welcome back. So we've just scratched the surface of this whole DNA revolution.
Right.
And now it's time to dive into some real-world applications of this crazy tech.
Sounds good.
Okay, so I'm really interested in gene therapy. You mentioned it earlier.
Yeah.
Can you give us a real-life example of how it's actually working?
Absolutely. One really amazing success story
Uh-huh.
is the treatment of severe combined immunodeficiency, or SCID.
Okay.
It's a genetic disorder that leaves babies incredibly vulnerable to infections.
It's hard to imagine.
Yeah.
Babies born with basically no immune system.
It is.
That's heartbreaking.
But gene therapy offers some hope.
Oh, good.
Doctors can actually use a modified virus as a delivery system. It's like a
cool.
microscopic Trojan horse to deliver a healthy copy of the faulty gene into the patient's bone marrow stem cells.
So it's kind of like replacing a broken part in a machine,
Exactly.
allowing the immune system to work like it should.
Yeah, and it's still early days for gene therapy,
Right.
but it's already having a huge impact for patients with SCID and other genetic diseases.
It's amazing to think that we're treating diseases at their source,
Yeah,
at the level of our genes.
right.
Speaking of genes, we have to talk about personal genome analysis.
Okay.
I'm hearing so much about it these days,
Yeah.
but I'm still not quite sure what it is.
It's basically like getting a personalized owner's manual for your body.
I like that.
So it involves sequencing a person's entire genome
Right.
to pinpoint any genetic variations that could make them more likely to develop certain diseases.
So instead of waiting for symptoms to show up,
Right.
you could identify those risks early and maybe prevent them.
It's about moving from reactive to proactive healthcare. Like, imagine if you knew that you had a higher chance of heart disease
Okay.
or some types of cancer.
Yeah.
With that knowledge, you could change your lifestyle, like
Right.
your diet and exercise,
Uh-huh.
or even start screenings earlier.
Yeah. That's a powerful idea.
It is.
Having a glimpse into your future health
Right.
and then taking control.
Exactly.
But I also imagine that all of this information could be really overwhelming
Sure.
or even scary for some people.
Yeah, you're right. It can be a lot to take in.
Yeah.
That's why genetic counseling is such a critical part of personal genome analysis. So trained counselors can help people make sense of their results,
Right.
weigh the risks and benefits,
Yeah.
and make informed decisions about their health.
So it's not just about the raw data.
Yeah.
It's about figuring out what it all means for you
Right.
and then how you can use that information to live a healthier life.
That's the goal.
Okay, well, let's shift gears a bit to another area where DNA technology is having a major impact, and that's forensics. I'm a total sucker for those crime shows.
I hear you.
So I'm always fascinated by how DNA evidence is used to solve cases.
It's like something straight out of CSI, right?
Exactly. Remember that PCR technique we were talking about earlier?
Oh, yeah.
That plays a big role in forensics.
It does.
So we're talking about amplifying tiny traces of DNA
Yeah.
found at a crime scene.
Exactly. Like, imagine a single hair
Okay.
or just one drop of blood.
Wow.
PCR can turn that tiny sample into a gold mine of information.
That's crazy.
It lets forensic scientists create a whole DNA profile
Right.
that can then be compared to suspects or even checked against a DNA database.
It's incredible to think that one tiny molecule can solve a crime.
It is.
And it's not even just about catching criminals.
Right.
DNA testing has also helped to clear innocent people
Yeah.
who were wrongly convicted.
It's amazing.
And speaking of solving mysteries,
Okay.
DNA technology is also being used to uncover our past, our ancestry.
Yeah.
I'm really curious about those ancestry kits that are everywhere these days.
Yeah.
Like, what's the deal with those?
Those kits are basically like a time machine for your family history.
Wow.
They analyze your DNA to figure out your genetic ancestry,
Okay.
tracing your lineage back hundreds or even thousands of years.
That's amazing.
Yeah, you can see where your ancestors came from,
Cool.
connect with distant relatives, and even uncover some surprising family secrets.
Oh, wow. That sounds like a lot of fun.
It is.
But I imagine there's some ethical concerns with this type of testing.
For sure.
Like what?
Privacy is a big one.
Okay.
You really need to understand how your genetic data is being used
Yeah.
and how it's protected.
Right. And then there are the emotional implications
Yeah,
of uncovering these unexpected family ties
absolutely.
or secrets.
Right. It can be a real emotional roller coaster.
So it's definitely not just about spitting in a tube and getting
Nah.
your results.
You've got to think about it beforehand and afterward.
Okay, that makes sense. It's a powerful tool
It is.
that can really change your life.
Yeah, for sure.
And it's not just about humans either.
That's right.
DNA technology can also be used to protect endangered species
Yeah.
and preserve biodiversity.
Exactly.
How does that work?
DNA analysis can help conservationists track animal populations,
Okay.
identify poaching hotspots,
Uh-huh.
and even figure out the genetic diversity of endangered species.
So it's like this genetic toolkit
Yeah.
for conservationists
Uh-huh.
that helps them make better decisions about how to protect these vulnerable species.
Exactly. It's another example of DNA technology being used for good
Right.
to help solve some of the biggest challenges facing our planet.
Well, I've covered a lot of ground so far,
We have.
from gene therapy to forensics to conservation.
Right.
It's clear that DNA technology is changing the game
It is.
in so many different fields.
And we're just beginning to see its true potential.
It's exciting.
It is.
Okay, well, speaking of pushing boundaries,
Yeah.
we need to get back to gene editing and all the ethical questions that come with it.
Sure.
We touched on it before,
We did.
but it's such a complex issue
It is.
that it really deserves its own conversation.
I agree.
Gene editing technologies like CRISPR,
Right.
they're so powerful
They are.
that we have this huge responsibility
Yeah.
to use them wisely.
Absolutely.
Okay, so let's dive into that ethical minefield in the next part of our deep dive.
Let's do it.
We'll explore the potential benefits and risks of gene editing
Right.
and really tackle some of the toughest questions facing science and society today.
It's a conversation we can't afford to ignore.
Right.
The choices we make now about gene editing will impact generations to come.
Welcome back to the Deep Dive. We've covered how scientists can decode and manipulate DNA.
Right.
And we've seen how it's being used in everything from treating diseases to solving crimes.
It's pretty amazing.
But now we're getting to the big one.
Okay.
The ethics of gene editing. It's a topic that brings up both hope and fear.
Yeah. It's a real double-edged sword.
I like that.
Gene editing technologies like CRISPR, they have this immense potential for good, but they also raise some really big ethical questions about what it means to be human and how far we should go in changing our genes.
Okay, so let's start with the good stuff.
Okay.
You mentioned earlier that CRISPR could be used to cure genetic diseases.
Yeah.
Can you give us an example of what that would actually look like?
Okay, so imagine a world where diseases like cystic fibrosis, sickle cell anemia, even Huntington's disease, they're no longer a life sentence.
Wow.
CRISPR could potentially fix the genes that cause these conditions, offering a cure where before there was only treatment.
So it's like erasing a typo in the code of life.
Exactly.
Giving people a chance at a healthy future they might never have thought possible.
Yeah. And the benefits go even beyond curing inherited diseases.
Really?
CRISPR could also help us develop new therapies to cancer, HIV, and so many other conditions.
The possibilities are pretty mind-boggling.
They are.
But there's always another side to the coin, right?
Of course.
Like, what are some of the ethical concerns that worry you?
Well, one of the biggest ones is unintended consequences.
Okay.
The human genome is incredibly complex.
Right.
And we're still figuring out how all these genes work together.
Yeah.
So even a tiny change to one gene could have ripple effects throughout the genome.
So it's like that butterfly effect.
Exactly.
Where one little change can lead to a ton of unexpected changes.
And then there's germline editing, which is when we make changes to the DNA in embryos or reproductive cells.
Right.
And those changes would be passed down to future generations.
Yeah, there's a whole other can of worms.
Right.
We're talking about changing the genes of future humans who don't get a say in it.
Exactly. It's a huge responsibility.
It is.
And we need to be incredibly careful.
Absolutely.
Where do we draw the line? Who gets to decide which traits are desirable and which aren't?
Those are big questions.
They are, and these are questions that society needs to grapple with before we go any further.
And you mentioned designer babies before.
Yeah.
Is that a real concern, or is that just sci-fi hype?
I think it's a valid concern.
Okay.
The technology might not be there yet to create babies with specific traits, but it's something we need to be ready for.
For sure.
The potential to choose things like intelligence, athletic ability, and even eye color.
Right.
It brings up some serious questions about human autonomy, equality, and what it means to be human.
It feels like a slippery slope.
It is.
Where does enhancement end and eugenics begin?
That's the big question.
Yeah.
We need open and honest conversations about this stuff.
I agree.
Involving scientists, ethicists, policymakers, and the public.
Yeah, this isn't just a decision for scientists.
No.
This is a decision for everyone.
Exactly.
So how do we navigate all of this?
Well, one important step is to create clear guidelines and regulations for gene editing research.
Makes sense.
We have to make sure this technology is used the right way, ethically, with proper safeguards in place.
But regulations can only go so far, right?
True.
We also need to build a culture of responsibility within the scientific community.
Absolutely. Scientists have to think about how their work will impact society and be open about the ethical implications of their research.
So it's about finding that balance between scientific progress and ethical responsibility.
Exactly. We can't forget that science is a tool.
Right.
And like any tool, it can be used for good or bad.
True.
It's up to us to make sure gene editing is used to make lives better, to create a healthier and more equitable future for all.
Well, this has been an amazing conversation.
It has.
We've gone from the basics of DNA to the ethical challenges of gene editing.
Yeah.
It's clear that DNA technology is a powerful force that's shaping our world in ways we're just starting to understand.
It's a journey of discovery that's both exciting and humbling.
I love that.
The more we learn about DNA, the more we realize how much we still don't know.
And with all this knowledge comes a lot of responsibility.
For sure.
It's up to all of us to make sure this technology is used wisely, ethically, and to benefit everyone.
I agree.
Well, thank you for joining us for this deep dive into the world of DNA technology.
It's been my pleasure.
And to our listener, keep those questions coming and never stop exploring the wonders of science.
Stay curious.
Until next time, keep on learning.